Hello, everyone, and welcome to today's webinar, Applications in Cancer Research, RNA Isolation from Organoids and Spheroids. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Laura Chapman, R&D Manager, Thermo Fisher Scientific. Anupriya Gupta, Scientist Level 2, Thermo Fisher Scientific. And Jay Bhandari, Scientist Level 2, Thermo Fisher Scientific. Laura, Anupriya, and Jay, you may now begin your presentation.
Thank you, and thanks everyone for joining today, and we're excited to present to you some of the research that we've been doing here at Thermo Fisher Scientific. To start off with, I just wanted to give a quick intro slide to our speakers today. So my name is Laura Chapman, and I'm an R&D manager, and I've been with Thermo Fisher Scientific for 16 years, so quite a while, and I've worked on several different things listed here under my picture. And today we're going to be focusing on spheroids and organoids. We also have Anupriya Gupta joining us, and she's been here for a few years and has some experience with our Kingfisher and Duo, Flex and Duo platforms for automation for sample prep. And then lastly is Jay Bhandari. He'll be presenting lastly. He's got extensive background in lots of cancer research, as well as here at Thermo Fisher, supporting our products and generating lots of data to highlight some of the applications for those. So thank you again for joining us. So this is a brief overview of our agenda for today. We'll give an introduction into cancer research, and then an introduction to organoids and spheroids, and then we'll move into some more of the data that we've generated here for RNA isolation from those 3D cell cultures using standard spin columns or Trisol, as well as some of our higher throughput using like the Kingfisher Flex, the Duo, and also a lysate-based method with cells-to-CT. So again, thanks for joining us. And I just wanted to give some high-level cancer statistics. The cancer is a burden worldwide. It's among the leading cause of death for the entire world. And just if you look at the cases from 2018 and then by 2040, it's almost double. So in about 20 years, we'll have almost double the number of cancer cases that are diagnosed. And so interestingly, I put on this third note, the cancer rates are highest among those countries that are well educated and have the highest life expectancy. However, some cancers are the opposite. In the poorer countries and such as cervical cancer, these are much higher rates in the poorer countries. And so it's quite interesting just due to differences in economic availability of medicines. So this is some data, high-level data for cancer and how it affects general population. This is specific to the United States. Interestingly, breast cancer is about 15% of the new cases diagnosed, as shown in the dark blue pie on the left side. However, if you look at that dark blue pie on the right side, it's only about 7% of the deaths. And this is due to the amount of research and early detection that's been encouraged for breast cancer diagnosis and early detection. Is the main thing is like preventing. And so you can see that with the amount of survival comparing the diagnosis versus the number of deaths. Again, if you look at prostate cancer, it's about 14% of the new cases that are diagnosed, as shown in the green. However, if you look at the deaths, it doesn't even show up in the deaths. So that's quite interesting as well. And again, it's because of early detection and the progress that we've made with researching these cancers. However, if you look at the lung and bronchitis cancers, those are 12% of the new cases, yet 21% die from bronchus and lung cancers. So this is highlighting an area where more research is needed. And then some other interesting data that I wanted to highlight. This is by gender, again in the United States, breaking down the different cancers that are diagnosed within males and females. If you look at the males, the #1 type of cancer that is diagnosed is prostate cancer. And then for females, the #1 diagnosed is breast cancer. And breast cancer is actually the #1 diagnosed of all cancers between males and females. Once you get past those, then it's pretty even. There's not a huge difference between genders as to which cancer diagnosis is received, except for uterine, which is primarily for females. So anyway, this is just highlighting some of the cancers that are top in the United States, but also need some more research. And some great links have been made in the research space, and some new ways of researching is using 3D cell culture. Some of you may be familiar with organoids and spheroids, but for those who are not, I wanted to give a brief introduction as to what 3D cell culture is and a definition of what we've been calling spheroids and organoids. There's a little bit of still discussion as to what an organoid or a spheroid actually is, and so this is the way that we've defined what an organoid and spheroid is. So spheroids are more simple. They are 3D cell aggregates. Generally, one cell type is all that is self-assembled within a cell culture vessel or some kind of environment. They can be grown in different ways using something like Matrigel or just in a non-adherent cell plate. And they like I said, they're typically only one cell type. Generally, you don't require scaffolding for spheroids, but you can put some Matrigel in the media, or you can have them grown on Matrigel dots. However, some people just have them grow in non-adherent plates. The other type of 3D culture that we'll be talking about today are organoids, and these are much more complex. They're 3D as well, multicellular generally. Different types of cells go into the structures that create these organoids. They are grown in vivo to mimic the parallel organ that would be found in vivo. They're grown in vitro to parallel the organ found in vivo. They can be derived from stem cells, either iPSCs or ESCs, and they generally take weeks to differentiate into those organoids. And today's data will be using midbrain organoids that were differentiated by some of our colleagues here at Thermo Fisher Scientific, starting from some ESCs that were differentiated down a path to create midbrain organoids. So that's the type of organoids that we used here. And then for spheroids, we had a few different cells that we used to create the spheroids for testing. And so with that, I'll pass it over to Anupriya, who will take over for the next section to present.
Thank you for the introduction, Laura. I just wanted to talk a little bit about 3D culture and add to some of the things that Laura has already mentioned. So like she said, they present an in vivo picture for something that is in vitro. So we're able to grow organoids or spheroids from primary tissues or immortalized cell lines, or and they resemble an in vivo pattern due to adhesion. That's their primary thing. Monolayer cells stick to the bottom of the plate, and 3D cell cultures, spheroids and organoids, generally stick to each other with adhesion. And the advantage over monolayer here we have is for cancer research, drug discovery, and toxicity testing. So here we are looking at some immortalized cell line spheroid generation that we grew in lab. These are HepG2 cells. These cells were diluted and plated in a 96 well plate, and from anywhere from 500 to 16,000 cells. And they were spun down after being plated, and that's where you see the day zero congregation of cells together. But as they start to grow in media, which was 10% FBS and DMEM, you can see that they clamp together pretty tightly and start to get really, really big and grow on top of each other in a 3D format. Again, these don't adhere to the bottom of the plate, they adhere to each other, and they're grown in non-adherent plates. So anywhere from 1 to 7 days, you can see a pretty linear growth. Here I'm going to mostly cover 4 kits that are commercially available on our website, and they kind of go left to right in the amount of the reduction in hands-on time and the increase in scalability / the amount of samples that you can run. So on the left here, we have Trisol. It's a phenol chloroform extraction, has a lot of yields a very, very pure and clean sample. Spin columns, everybody uses them. Everybody's familiar with them. It works with a filter and you centrifuge your sample down. Crude lysate, which is direct lysis into qPCR, only 7 minutes of lysis time. And then magnetic beads, which can be put on a instrument so that you do some hands-on processing early on, and then you put all the downstream wash steps and elution onto a machine, and that's mag bead based. Trisol through Nirvana, all of these kits are available on our website. So let's talk a little bit about Trisol and Purelink. These 2 kits have been around for decades, and people use them all the time, and there's familiar results, and it's consistent in its purity. But the only issue is that with Trisol, you have some hazardous material, so you need to work in the hood, and a high hands-on time. So you're sitting by the centrifuge and watching your samples spin down, and it's generally low throughput due to the fact that it is very high hands-on time. All right, let's look at some data concerning spheroids and organoids when extracted with the Trisol kit. So these spheroids that you're seeing on the top left in the picture, they were grown for 48 hours. They're HepG2 spheroids, and after 2 washes with PBS, once the media was removed, 25 microliters of PBS was left prior to extraction. When you look at the chart on the bottom, there's average CTs on the y-axis and the number of seeded cells per spheroid on the x-axis. So we're going from 500 to 16,000. And you can see that there is quite a bit of variability with the Trisol extraction. We may have possibly lost a little bit of cellular material looking at the 2,000 and the 16,000 data sets. And that's why you see a lot of variability in the error bars. For organoids on the right, there's average CD on y-axis and the number of samples on the x-axis. So the left bars are Trisol and the right bars, red and black, are 8 organoids pulled together and then extracted. And you can see that there is about a 3.3 CT difference when you pull 8 organoids together. So we can see linear recovery. On the bottom, it's an Agilent trace. Agilent that's an electropherogram, and you can see that the Trisol gives pretty good RIN values at 9.4. Now we're going to look at some Purelink spin column RNA extractions. So again, these are 48 hours of HepG2 spheroids left with 25 microliters of PBS left in the well, and you can see a pretty significant linearity in the number of seeded cells going from 500 to 16,000. That's average CTs on the y-axis and seeded cells on the x-axis. And on the right, that's a representative picture of an iPSC spheroid. And the orange bars show that we added a xeno as a positive control internally, and you can see that there is no xeno inhibition. Therefore, all of these samples are pure and without inhibition. Looking at the bottom graph, we compared the extractions of 1 and 2 spheroids. So we wanted to see if there was linear recovery. And you can see that it's about a CD shift downwards to 2 spheroids, and that also indicates linearity. Here are some of the organoids. These are midbrain organoids that were grown for 6 weeks in suspension while shaking, and then they were shipped to us overnight in hibernate medium, but they were not frozen. You can see that there are pretty significant differences in morphology, and because these organoids are more tissue-like, they're very tightly bound and it's pretty difficult to lyse them. So whatever our standard method of lysis is, we had to add 2 or 3 minutes extra on the vortexer and maybe a little bit of pipetting to make sure that we were able to get efficient lysis. On the bottom with the blue graphs, with the blue bars, you can see that that's average CT on the y-axis and 3 housekeeping genes on the x-axis, CDK, GNE, and GPI. And we can see pretty good CTs with the Purelink extraction. The RIN values for Purelink on the Agilent trace is a little bit lower at 6.9, but still pretty decent recovery and quality. And with that, I will pass this over to Jay.
Thank you, Anupriya. That was a really good intro to our gold standard methods for RNA isolation using spheroids and organoids. Again, my name is Jay Bhandari. I'm one of the application scientists here at Thermo Fisher Scientific. In the next few slides, I'll introduce some of the automated and high throughput extraction methods for these 3D cultures and present some experimental data for RNA isolation for spheroids and organoids using these methods. So our automated extraction methods utilize magnetic bead-based purification. So magnetic beads offer many benefits compared to traditional methods for isolating total RNA compared to any other organic extraction using Trisol that Anupriya mentioned earlier, or spin column-based extraction with Purelink RNA mini. So magnetic beads bind RNA, which enables higher and more consistent yields and more reproducible data. Let me begin by introducing our MagMax Nirvana Total RNA Isolation Kit. The kit uses magnetic bead-based technology and is designed for the isolation of total RNA, including small RNAs, such as microRNAs, from a wide variety of sample types. The kit is optimized for use with multiple sample types, which could be serum, plasma, whole blood, tissue, cell culture, and urine. In other words, there is no need to order special kits for each of the sample types. So one kit for the diverse set of samples. So with the magnetic bead-based purification, you can scale from processing 6 to 96 samples at once when combined with the Kingfisher instruments, which could be Kingfisher Duo, Kingfisher Duo Prime, Kingfisher Flex, or Kingfisher Apex. So alternatively, you can also use or you can process your samples manually with the magnetic stand. So in terms of workflow, magnetic bead-based extraction can be divided into 3 basic steps. So prepare plates with samples and reagents, typical pre-processing steps, run a program on a Kingfisher instrument. The instrument can be either of those, like I discussed earlier, which can run, like Kingfisher Duo, which can run up to 24 samples at once, or Kingfisher Apex, or Kingfisher Flex, that can actually run up to 96 samples. After you run a program, then you collect the total RNA at the end of the run, which could depending on your sample type, it could either be 60 minutes, 75 minutes, or either 1.5 hours. And the benefit of using automated extraction is that it reduces your hands-on time significantly compared to more traditional methods like Trisol or Purelink. To put that into perspective, with Purelink and Trisol, it will probably take you about 2 or 3 hours to process the samples from start to finish, and you will be constantly working on the bench. With magnetic beads and Kingfisher automation, your hands-on time is significantly reduced. The total turnaround time is for cells is about 75 minutes, but the total hands-on time for pre-processing and setting up the plates is only about 20 minutes. For tissues, the total turnaround time is about 90 minutes, but the total hands-on time is approximately about 35 minutes. So automation definitely helps reduce your hands-on time. Let's look at RNA isolation from different spheroids using this MagMax Nirvana tool RNA isolation kit. So on the top left here, you can see the spheroids from Hep G2 cells. The spheroids were formed by the addition of various number of cell inputs, shown here on the top and grown for 48 hours. The scale bars on the bottom shows the average diameter of the spheroids from different hepatitis cell inputs after 48 hours of growth. So we took these individual spheroids formed on a 96-well plate, washed them with PBS, and used the cell lysis and binding protocol from the MagMax Nirvana total RNA isolation kit. And at the end of the run on the Kingfisher instrument, we do get a clean RNA. And the eluted RNA was used to compare gene expression of some of the common housekeeping genes, like the kinases mentioned here, using the RT-PCR. The graph here shows the average cycling threshold, or CT values on y-axis, and number of cells seeded per spheroid on the x-axis. A single spheroid was used for extraction in this experiment, and we can see that there is a linear recovery of CT values from the spheroid generated from different cell numbers. On the top right, you can see we looked at spheroids generated from induced pluripotent stem cells, or iPSCs. These were significantly larger in size compared to Hep G2 cells, Hep G2 spheroids on the left. We performed an extraction using a MagMax Nirvana kit and looked at gene expression profiles from these spheroids. We also included an internal positive control, or the GenoRNA spiking control, to compare the performance of the kit and check for RNA inhibition from these samples. And we saw the expected CT values for geno expression, suggesting the absence of inhibition from RNA isolated from these iPSC spheroids. So this suggests that MagMax Nirvana kit can efficiently be used to isolate spheroids from iPSCs and also from Hep G2 cultured cells. We then compared efficiency of the kit for multiple spheroids. Basically, we asked a couple of questions. So one, can we apply MagMax Nirvana for RNA isolation from multiple spheroids? Or two, do we get consistent recovery of RNA? And the answer to both is yes. You can use MagMax Nirvana for RNA isolation from two spheroids, and we do get linear recovery of gene expression. And for these experiments, we actually used cell input of 5,000 cells per spheroid. And as expected, the CT difference for these gene targets expressed in individual spheroids versus a pool of two spheroids was approximately about one cycle. To summarize our findings, we say that our MagMax total RNA isolation kit was able to efficiently isolate RNA from spheroids for gene expression studies. Next, we also looked at RNA isolation from organoids. So organoids, like another organ-like 3D cell culture that my colleagues actually talked about earlier. So we used human midbrain organoids. Again, these organoids are slightly larger in size. They have some strict growth requirements. So we tested these organoids with the kit, the MagMax Nirvana kit chemistry. To begin with, we found that the pre-processing steps, mainly the lysis step in particular, is a little challenging for these sample types. So we did have to modify the recommended protocol to enable complete lysis of these organoids. We increased the lysis time to 2 to 3 minutes on a vortexer to get complete lysis, and rest of the protocol was similar to that used for RNA isolation from spheroids. So these isolated RNA was used to study gene expression, and as you can see, we do get good CT values for the housekeeping genes shown here on the bottom left. So one thing of note here is that we did see difference in gene expression profile from human midbrain organoids, which were cultured using two different growth conditions. So there is always, it is important to optimize your RNA isolation, which is suited suitable for the different 3D cell culture that your lab is working on, suitable for your growth conditions and your experimental needs. To ensure that the extraction protocols produced high-quality RNA and did not reduce RNA integrity, we also analyzed the quality of RNA isolated from individual organoids using the Agilent bioanalyzer. So the electropherogram traces indicated that high-quality RNA could be isolated with minimal loss of integrity, as shown here by Win value or RNA integrity number of 8.7. So the total RNA, including some small RNAs, was more efficiently isolated with the MagMax chemistry, and then the isolation here in the purple box highlights significant recovery of small RNAs. To conclude, we can say that automated extraction using magnetic bead-based MagMax Nirvana total RNA isolation kit is efficient for RNA isolation from these 3D cultures and significantly reduces your hands-on time. So in the next section of the presentation, I'll switch my topic and present an efficient high-throughput RNA isolation from spheroids and organoids. Mainly, I'll introduce Cells-to-CT. Basically, Cells-to-CT kit allows us to study gene expression by real-time qPCR without RNA purifications. These are available in both Chapman and CyberGreen detection chemistries. So these kits provide a complete workflow for gene expression studies directly from cultured cells, providing a user with excellent performance and also reliable results. In terms of the total time, the sample prep time is only about 7 minutes for these kits. In terms of workflow, once you have your cultured cells, wash them with PBS, prepare lysis solution with DNase, add your lysis solution to the cells, pipette and mix 5 times, and then incubate the cells for 5 minutes. Then you add your stop solution and pipette mix 5 times and incubate at room temperature for 2 additional minutes. And from this stage, you could choose one of the 2 steps for processing samples. You could do a 2-step workflow where you can take your lysate, reverse transcribe to get cDNA, and use that cDNA to do quantitative PCR to study gene expression. Or you could also follow a 1-step workflow where you can go directly from your cell lysate into a qPCR for gene expression study. So both the RT and qPCR are performed in a single step. So how do you decide which workflow to choose depends on your research needs. So you can choose 1 step if you have many sample types if you have many samples with 1 or few targets, and if you do not like to store your cDNA. But you would probably like the 2-step workflow if you have multiple targets quantified from cDNA and need higher sensitivity for rare transcripts, and also you archive your cDNA. So we then looked at RNA isolation from spheroids and organoids using this same workflow, and for the experiments presented here, we performed a 1-step workflow to look at gene expression studies. So again, images of the Hep G2 spheroids were grown for 48 hours, like we did talk about earlier, with various cell input. And you can see that the average CT values indicated linear recovery of RNA from Hep G2 spheroids. We also looked at spheroids generated from iPSCs, and these were significantly larger compared to Hep G2 on the left. We performed an extraction using Cells-to-CT kit and looked at gene expression profile from these spheroids. We also included the GeoRNA spike-in control, and we do see no RNA inhibition from these samples, and we do get expected CT values for gene expression. We then compared the efficiency of the kit for multiple spheroids, and as expected, you do see a difference of 1 cycle between 1 spheroids versus 2 spheroids grown with same cell input. So we then looked at RNA isolation from primary hepatocyte spheroids. So the average diameter of these primary hepatocyte spheroids, which were grown from 1,500 cells for 5 days, was approximately around 300 microns, and these are slightly smaller. The lysis efficiency of the Cells-to-CT kit actually depended on how much PBS can be removed after washing the spheroids. So to test the effect of PBS volume on isolation of RNA and gene expression, we extracted RNA from these spheroid samples with various amounts of PBS remaining on the plate, and then the average CT values we recover were lower but less variable when the initial samples contain lower volume of PBS, such as 10 microliter. But if you can remove all the PBS, that will probably give you better gene expression profile for your spheroids. So this actually suggested that the larger volume of PBS probably is influencing your lysis efficiency. Therefore, removal of excess PBS is critical for gene expression studies from these 3D cultures. Also, the smaller size of the primary hepatocyte cultures may have contributed to additional variability in extraction efficiency, just because these were difficult to work with. Like you cannot keep them intact while taking your PBS out of the plate. So remove the PBS as much as you can just to get better expression profile, but be careful that you're not losing your spheroid at the same time. And also, this would also suggest that pooling smaller size spheroid cultures may minimize heterogeneity and produce consistent recovery from these spheroids. On the graphs here on the bottom right, you can see that there is a difference in gene expression profiles for primary hepatocytes taken from 3 independent donors, suggesting there is donor-to-donor variability for gene expression as well. So it's critical to choose your primary hepatocytes at the beginning to work for your downstream analysis. We then looked at organoid isolation using our Cells-to-CT kit. Again, these were human midbrain organoids and a little challenging to work with. For these experiments, we used single organoids. For these organoids, in order to achieve complete lysis, again, the samples had to be vortex for 2 to 3 minutes, and the lysis time was extended from 5 minutes to 7 or 8 minutes in order to achieve complete lysis of the samples. On the graphs here at the bottom, you can see good gene expression of housekeeping genes from these organoids. What is not shown here is the RNA inhibition from these sample types using GenoRNA spiking controls. Basically, there was no inhibition, RNA inhibition from these sample types. So we can say that Cells-to-CT can be used for RNA isolation from these cultures, but you do need to modify some of the recommended protocol for this and optimize experiments for your research needs. And up to this point, I have talked about each of these methods in isolation, but how do they compare? So how do I decide which ones to choose? What is the experimental data against of these kits? So in order to compare the efficiency of each of the methods for RNA isolation, we isolated RNA from these samples side by side. And here you can see on the graph on the left, you do get consistent recovery of RNA and gene expression from spin column-based Purelink RNA mini and also the magnetic bead-based MagMax Nirvana, shown here by consistent recovery of the average CT value. Cells-to-CT, so higher CT values for low cell input, but showed consistent recovery compared to Purelink and MagMax. Nirvana for high cell input spheroids. Organic-based Trisol extraction did recover good RNA, but was more variable and less consistent out of the 4 methods we tested. So for RNA isolation, compared between 4 extraction methods from human midbrain organoids, there was a consistent recovery of gene expression from all the 4 methods that we tested. However, I do want to point out here that modifications of the workflow had to be done to get complete lysis of these 3D cultures. However, our results show that with slight modification, you can actually get RNA recovery from 3D cultures using any of the 4 methods. It is also critical to completely remove PBS from the samples to increase lysis efficiency, particularly for cells-to-CT. And also, again, deciding on which methods you should choose for your research needs, it also depends on what your downstream analysis would be. So here, what I would like to say or point out is, how do you choose a product for your research? So how do you decide which ones to go with? So basically, selection of an appropriate extraction method actually depends on any end-user requirements and also a particular experiment and whether a manual or low throughput or a semi-automated workflow is desired. So right off the bat, cells-to-CT would be ideal for high throughput users who need to process individual spheroid and organoid samples for gene expression studies, but you do need to modify your protocol according to your needs. Purelink Mini Kit or Trisol might be preferable to users who process less than 24 samples at once. And MagMax Nirvana Total RNA Isolation Kit, which is a magnetic bead-based extraction, will be compatible with high throughput purification of RNA and also DNA. So it is favored by end users with NGS and qPCR workflow. Extraction with Trisol may be desired if you're pooling multiple spheroids or organoids, or have to be pulled, like if you need to get a really good gene expression profile from small spheroids. And again, each of the methods do actually come with really good RNA extraction or good quality RNA for your downstream assays, depending on what your needs are. So next, I'll briefly talk about the microRNA analysis using our cells-to-CT kit. This may also be relevant to some of our audience in the call today. MicroRNAs are small non-coding RNA molecules that play major roles in post-transcriptional regulation of gene expression. And there is always an interest in researching microRNAs because of their association with cancer. So in order to study microRNA from cultured cells, again, the experiments present here is with cultured cells and not with the 3D cultures. We can use cell lysate from cells-to-CT, just like I talked about earlier for 3D cultures. The only modification was that we increased the lysis time from 5 minutes to 8 minutes to get complete lysis and recovery of small RNAs or microRNAs. Once you get the lysate, you can use Chapman microRNA to reverse transcribe single microRNA or use advanced microRNA assay to reverse transcribe multiple microRNAs using universal reverse transcription primers, which will then go into your qPCR reaction for microRNA expression studies. So here you can see on the 2 graphs here on the right, with 2 different cell types, you do get accurate and robust measurement of microRNA, which can be achieved with any of the 2 workflows you choose to study microRNA with. So if you would like to learn more about the methods we discussed for RNA isolation from spheroids and organoids, please visit our website for additional resources. We also have an application note for studying microRNA. How do you go about studying microRNA from cultured cells using cells-to-CT? Furthermore, there are additional resources on the web for cells-to-CT with links to how-to videos, previous webinars, presentations, tips and tricks, application notes, technical bulletin, flyers, and much more. With that, I'd like to thank everybody who joined this webinar today, and thank you very much for your participation. We would take any questions that you may have. Thank you, everyone.
Thank you, Laura, Anupriya, and Jay for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, which method would you recommend for pooling multiple organoids?
I'll take a quick stab at that. So for pooling, since it's a larger number of samples and a larger, mass of cells, you would want something that has a little bit stronger lysis. So you wouldn't want to use the cells-to-CT. That's going to be better for smaller sample input. So Trisol is a good option for low throughput, high purity if you want to do like RNA sequencing and qPCR downstream. You'll have really pure RNA to be able to do any of your downstream analysis. You could also use the spin column method with the Purelink RNA spin columns, or even the MagMax Mirvana. However, that one has a smaller lysis volume, so it would depend on the amount that you want to pool, whether that would work very well or not. Probably, if it's a larger number of samples, you would want to use either Trisol or the spin column Purelink RNA total kit. If it's a smaller number of one or two, then you could, maybe even up to 5, depending on the total size. You could use the MagMax Mirvana kit if you wanted a higher throughput solution.
All right, thank you. Next question, which method would you recommend for screening 96 well plates?
I can take that. So first of all, with the spheroids that you make, you typically tend to use 96 well plate, but you can also do it in a larger batch in a flask. If you're using a 96 well plate to grow your spheroids, we would recommend either the MagMax Mirvana for magnetic bead-based extraction, or you could also go with the cells-to-CT kit, which offers you a sample to solution and has like high throughput options. Would be able to screen for single spheroids grown on a 96 well plate. So it kind of depends on whether you want to get your results sooner or whether you have the capacity to do automated extraction using the magnetic bead-based extraction. Either of the two would be a good fit for those.
All right, and we will wrap up with that question. Thank you, Laura, Anupriya, and Jay. Do you have any final comments for our audience?
We'd just like to say thank you for your time. We've really enjoyed sharing some of the research that we've done here. If you have any other questions, feel free to reach out and send those along, and we can get back to you with answers. But thank you for your time. We've really appreciated and enjoyed sharing it with you.
Thank you.
Thank you.
Thank you again, Laura, Anupriya, and Jay, for your time today and your wonderful presentation. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.